This is my YouTube channel, Biological Discussion. I am Shak Banerjee. I want to discuss biology with you through this channel to gather more and more knowledge about this subject. So let's start today's topic. After hearing the term biology, the first word should come on our mind is cell. We all know what is cell, the basic structural and functional unit of cellular organisms. It is an aqueous compartment bound by cell membrane, which is made up of phospholipid bilayer and which is capable of independent existence and performing the essential functions of life. All organisms more complex than viruses because they consist of cells. Viruses are non-cellular organisms because they lack cell or cell-like structure. In the year of 1665, Robert Hooke first discovered cell from a piece of cork and also coined the word cell. The word cell is derived from the Latin word cellula, which means small compartment. Hooke published his findings in his famous work Micrographia. Actually, Hooke only observed cell walls because cork cells are dead cells and they are without cytoplasmic content. Anton van Leeuwenhoek was the first person who observed living cells under the microscope and named them animalcules. It means little animals. On the basis of internal architecture, all cells can be subdivided into two major classes, prokaryotic cells and eukaryotic cells. Cells that have unit membrane bound nuclei are called eukaryote, whereas cells that lack a membrane bound nucleus are prokaryote. Cells control our body by playing different types of roles. One of them is cell signaling. In one line, cell signaling is cell communication. All cells receive and respond to signals from their surroundings. This is accomplished by a variety of signaling molecules that are secreted or expressed on the surface of one cell and bind to receptors expressed by another cells, thereby integrating and coordinating the functions of the many individual cells that make up organisms. Each cell is programmed to respond to specific extracellular signal molecules. Signaling by direct cell-cell or cell matrix interactions plays a critical role in regulating the behavior of cells in animal tissue. For example, the integrins and cadherins function not only as cell adhesion molecules but also as signaling molecules that bind to adjacent cells or the extracellular matrix and regulate cell proliferation and survival in response to cell-cell and cell matrix contacts. Here you should know that integrins are proteins that function mechanically by attaching the cytoskeleton, and cadherins are a type of cell adhesion molecules that is important in the formation adhesions junction to bind cell each other. Cell adhesion is the process by which cells form contacts between each other. In addition, cells express a variety of cell surface receptors that interact with signaling molecules on the surface of neighboring cells. Signaling via such direct cell-cell interactions play a critical role in regulating the many interactions between different types of cells that takes place during embryonic development as well as in the maintenance of adult tissue. Cell signaling are mainly two types: extracellular signaling and receptor associated signaling. Extracellular signaling usually involves the following steps: 1. Synthesis and release of the signaling molecule by the signaling cell. 2. Transport of the signal to target cell. 3. Binding of the signal by a specific receptor leading to its activation. And 4. Initiation of signal transduction pathway. In animal, extracellular signaling by signaling molecules can be classified into 4 categories: endocrine, paracrine, autocrine, and juxtacrine. In endocrine signaling, the signaling molecules act on target cells distinctly located from their site of synthesis. It is a long-range signaling in which signal molecules is transported by the bloodstream. The signaling molecules, such as hormones, neurotransmitters, are secreted by specialized endocrine cells and carried through the circulation to act on target cells at distant body sites. A classic example is provided by the steroid hormone estrogen, which is produced by the ovaries and stimulates development and maintenance of the female reproductive system and secondary sex characteristics. In animals, more than 50 different hormones are produced by endocrine glands, including the pituitary, thyroid, parathyroid, pancreas, adrenal, and gonads. Next is paracrine signaling. In paracrine signaling, the signaling molecules released by a cell affect target cells only in close proximity. An example of this is the action of neurotransmitter in carrying signals between nerve cells at a synapse. In this signaling, the signaling molecule does not carried by the circulation system, rather than it diffuses in the extracellular matrix and attaches with the receptor of the target cell. Here you can see the neurotransmitter is released from the presynaptic cells, which is a nerve cells, and diffuses into the synaptic cleft. After that, it attaches to the receptor of the postsynaptic cell. Here the presynaptic cell is the signaling molecule synthesizing cell, and the postsynaptic cell is a target cell. So neurotransmitter is signaling molecule and attached with the receptor of the postsynaptic cell. So it is a good example of paracrine signaling. Next is autocrine signaling. In autocrine signaling, the signaling molecules produce an effect on same cell that produces it. Again, I say you that the signaling molecules produce an effect on same cell that produces the signaling molecule. One important example of such is the response of cells of the vertebrate immune system to foreign antigen. Certain types of T lymphocytes respond to antigenic stimulation by synthesizing a growth factor that drives their own proliferation, thereby increasing the number of responsive T lymphocytes and amplifying the immune system. It is also noteworthy that abnormal autocrine signaling frequently contributes to the uncontrolled growth of cancer cells. In this situation, a cancer cell produces a growth factor to which it also responds, thereby continuously driving its own unregulated proliferation. In the picture you can see that the cell molecules produces on an effect the same cells that produce it. And at last, in juxtacrine signaling, signaling molecules do not diffuse from the cell producing it, and cell-bearing signaling molecules interact with receptor proteins of adjacent responding cells. Unlike other modes of cell signaling, juxtacrine signaling requires physical contact between the cells involved, Notch signaling and classical cadherin signaling are examples of juxtacrine signaling. Here in the picture you can see the first cell that produce signaling molecules are embedded with the cell membrane, and the adjacent cell's receptor attach with the signaling molecule through a physical contact. So here it is clear that a physical contact is necessary between the cells involved in this signaling. So friend, so for today, thank you for watching my video. Next class we will discuss about intracellular signaling. Please motivate me to prepare more and more video, and like, share and comment this video and subscribe my channel. Thank you.